Good morning, everybody. Today, my topic is high throughput screening by machine learning application for drug discovery and computational modeling for SARS-CoV-2 main protease inhibitors. As COVID-19 persisted, the mutation of virus challenged available treatment, and currently only 3 antivirals are approved, and only one specifically designed to target the COVID-19 on the material. However, the efficacy and the accessibility of these drugs still limited. So this underlying the urgent needs of new antiviral drugs. So this is where SARS-CoV-2 main protease become a promising target due to their high conserve, which means they reduce the resistance risk, and also with it's also play crucial role in the viral replication, and ultimately their unique substrate specificity make it minimize the likelihood of inhibitor will cause react with the human enzyme. So to accelerate the Mpro inhibitors discovery, in silico drug design were used in this research and commonly rely on molecular docking, which is computationally expensive, time consuming, and there's also so we use the apply the machine learning to overcome these challenges and enable us to screen millions of compounds and enhancing the success rate. So this research utilized the machine learning to identify novel SARS-CoV-2 main protease inhibitor through an in silico pipeline for the virtual screening. And for our methodologies, we begin with the obtain the half maximum inhibitory concentration of IC50 of each molecule to SARS-CoV-2 main protease from available assay from the ChemBL database. And then the second, the chemical feature of those we will encode using parallel descriptor. And the several machine learning model were built to predict the IC50 of each molecules. And then we prepare the chemical library containing 1 million compound adhering to molecular Lipinski's rule of 5 from the ChemBL database. And we use the base model to screen those compounds, and we get the active compound. And after that, we prepare the ligand optimized and prepare the protein structure of main protease by proteination adjustment and put it in together to molecular docking analysis. And after we get the hit candidate, it will be evaluate the pharmacokinetic and pharmacodynamic for favorable properties that we need. So for our discussion and result and discussion, from the data pre-processing process, we received total of 785 molecules were selected for the model building after the assay screening from the database. And for the model prediction, our performance of the model, random forest give us achieve the highest performance with 95% accuracy and in training set and 91% accuracy in testing set. And the QQ part show that the predictive value has strong agreement to the experimental data with R square equal to 0.7. And also, according to the model, we identify important feature. Presence of oxygen and aromatic link in molecules were crucial for the inhibitory effect. And the final random forest model, after we develop some more, we apply for the screening about 1 million compounds, and we received that 15,000 molecules were classified as active, as the predicted value of IC50 lower than 100 nanomolar. After that, we do a molecular docking to validate our result from the machine learning. From the docking, we identify 17 potential inhibitors. And this is the example of 3 inhibitors that we have. And for the hydrogen bonds were the main non-covalent interaction and mostly involved in the residues such as serine 144, histidine 163, glutamate 166, and cysteine 145. With this cysteine 145 play a central role in enzymatic activity. So it's suggesting that the identified molecule potentially inhibit these key residues. And note that amide motif recurrent in top-line compound. So it's suggesting that the potential role of this motif in Mpro inhibitors. And this result indeed support by the evidence that large number of amide group was usually introduced for development of new broad spectrum antiviral, which support our work. And for the pharmacokinetic and pharmacodynamic analysis, from the boy-egg pot revival that one compound of our 17 have ability to cross the blood-brain barrier, and 5 of them have a low GI absorption, which need to be concerned. And luckily, the top 3 did not cross the blood-brain barrier and have high GI absorption. So to conclude our work, this study highlight the power of machine learning in accelerating the drug discovery identified 17 potential SARS-CoV-2 main inhibitors with favorable properties, offering a scalable framework for fighting future emerging disease, and it's also identified the critical Mpro residue for hydrogen bond formation, guiding the future of COVID-19 drug development. Thank you for your attention. Thank you.
Thank you. Do you have any questions or comments?
You got a large number of molecules that have the potential inhibitor, and I would like to know what's the criteria you select the candidate.
From the predicted value from that machine learning has predict less than 100 nanomolar.
The value.
Value of 100 IC50 less than 100 nanomolars. This you mean this number or?
And for the machine learning, what are the criteria?
For
you select only the 17 molecule because you got a large number of the molecule and how to?
I mean, we got 15,000 molecule for machine learning, and we select all of those 15,000 molecule into molecular docking to validate the best inhibitor. Like we use the material that the recent developed from the Pfizer as the baseline to see which molecule have higher binding score than this to identify the highest potentials, which also validate the machine learning.
According to this is in silico, right? So and it's based on the machine learning, and as we know that all of that developed nowadays, even though we use a lot of technology and we select by human, and we have already select some type of the medicine that is a candidate one for working, and finally after many try, most of them disseminate because of their not success. And how you sure that by training of your machine it will give a better result by compared to the human that we work for many years?
For the machine learning, I mean, when we as a people screening it more like we just found some molecules and we think that this might have an opportunity to be the top candidate. However, if you imagine in the hills, we ask some traveler where they are, and we think he is on the top of the hill. But machine learning can picture all of that hill and see where is the best molecule or who is on the top of the hill. So that is the benefit of machine learning that can model all of the property and which drug is the best. So and also there is many proof that using machine learning comparing to human have a higher success rate. At literature review state that around 10% to 15%. And also our molecular feature that we get also correlate with the property that can forming many hydrogen bonds, which means our machine learning also validated.
Thank you for the presentations and the great topics.